All right, so we're talking about uh, three things, one of which you guys don't have to worry about, which is negative staining. You guys don't have to do it. So, hey, one less thing to do, right? All right. Um, so, with this, we're going to talk about our cool science thing. So, gram staining has been around for almost hundreds of years. I think it's like 160 years. It's been around a long time. The proper gram staining proper that we have, where we differentiate between gram positive and gram negative bacteria, has been around a little bit more recently, but this helps differentiate bacteria based off of their cell wall properties. If you're gram positive, hopefully you guys know that they have a thick protein matrix, really, really thick. They have a single membrane, which is a cytoplasmic membrane. Gram negatives have a dual membrane. They have an outer membrane and an inner membrane. And in between, they have a small, tiny little bit of protein matrix and a bunch of other stuff hanging out around them. They've got a little bit more complexity in that sense. I'm thinking about it. I should turn off the phone so it doesn't ring. Great. Now, with a lot of things in science, you'll find that people go, hey, this is how something works, or this is how we think it works. We don't really have a way to test it. And so we just keep saying this is how it works, and then nobody gets around to testing it. And that happens a lot. You'd be surprised how many times this happens. But gram staining is one of these things where people are just like, well, it works. It generally works. It's pretty consistent. But we kind of have been telling you guys, hey, this is how gram staining works in textbooks and stuff for years. Might not actually work this way. These guys did a little bit of research and found out some interesting things. So how we assume gram staining works currently is we say, hey, we put in a bunch of dye, crystal violet dye. We let it sit in there for a little bit. We put in some mordant, which helps make these crystals. And these crystals get trapped in here on the inside of the gram negative and the gram positive. And then we wash it with alcohol. And the alcohol takes everything out of the inside of the gram negative cell. But because there's a protein matrix on the outside of the gram positive, it just stays in. So they're like, hey, well, let's test this out. It's some E. coli. So these guys grabbed some E. coli, and they were like, let's do some testing. And they use two types of testing, which are secondary harmonic signaling and transmembranal analysis. You don't need to remember that. It's really complex. And they're chemists, so they don't know anything about biology. So just ignore that part. But what they found when they were looking at this, as you can see, here's crystal violet dye they exposed to a cell. And they counted how long it took until it got to a saturation point. And it took only about 200 seconds. And they took a second molecule, which is called malachite green. And the only difference between these two is this little R group here. Malachite green just has a hydrogen. Crystal violet has an N-methyl-2, which is a pretty much bigger molecule. Not that much bigger, but pretty big. The malachite green took 500 seconds to saturate. Well, that's weird. They're almost the same shape. Why did they take such a big difference in time? You'd think the malachite green would go faster, right? Smaller molecule goes in faster. So they did these tests, and what they found with the saturation points, that there's a huge time gap, especially for the malachite green. And the malachite green has this weird dip. So they looked at it on the second generation harmonics signaling, and what you see is this inset part is this part right here. So this inset part is this guy right here. And for the crystal violet, you can see this one really quick, big spike in the beginning, and then it just kind of levels off. It just levels off throughout the peak. But for the green one, you see a big spike in the beginning, and then this secondary spike right here, and then it levels off. And what this means is that the malachite green is going through the first layer of the gram negative, chilling out in this protein matrix, and then going through into the interior of the cell. And it does that at a slightly different rate. It goes from here to here pretty fast, and then from here to here very fast. Whereas the crystal violet just kind of crosses the first one, and it just kind of chills out in this area. It doesn't actually cross into the gram negative. So long story short, we've been lying to you guys about how this works. It probably says it in your textbook the other way. But here's the thing. This is one paper. As far as I can tell, nobody kind of did follow-up research, just verify, hey, let's double check these things. But basically, these guys were like, what's probably happening is this, which is the crystal violet goes in between the gram negative layers, and it kind of saturates inside the gram positive, When you put in the mordant, it makes the crystals in the gram-positive and in the gram-negative, and then you do the alcohol wash. And everything kind of gets trapped inside the gram-positives, and the reason why is because it has that thick protein matrix, and when you expose protein matrices to alcohol, they kind of melts up. They just kind of glom together and hold on to things really tightly because they're denatured. And in this case, it's holding in the dye. And because the alcohol also causes membranes to lyse, then the dye is held on the interior of the cell, so it's really easy to visualize. Whereas the gram-negative, because it has that thin protein matrix, the protein matrix just kind of lets it go, and it strips out the outer membrane, so the cell doesn't hold on to anything. So, basically, what we did is we found out that we just really didn't understand this fully. It took us a really long time to kind of figure this out, but yeah, it turns out it's probably not the way we tell you. But we're still going to tell you that way because we don't know for certain it's this way. But there's pretty good evidence that there is. So, cool science thing of the week, take home message. Gram staining is probably not what we think it is, and it doesn't actually go on the interior of gram negatives. Fun facts. Okay, so we're going to talk about staining. Safety stuff first. If a slide has bacteria on it and hasn't been heat fixed, and it cracks or something like that, make sure you notify your instructor right away, and we'll help you decontaminate the working area. Heat fixing does kill bacteria, so once it's heat fixed, then it's fine. But if it's not heat fixed, then it's not. Goggles, gloves, and lab coat must be worn at all times, except for when you're looking through the microscope. So once you guys are done with all the bacterial handling, done with all the staining, you sit down to use microscopes, you can take your gloves off and use the microscopes and your goggles. So also, don't wear your gloves while you're touching the microscopes. Gloves tend to have dye on them. We don't want to put dye on the microscopes because it's shiny and new and white, so that makes it really evident that you put dye on there. So don't do that. It's really hard to get off. Also, please be very careful not to spill dye on the floor. It is a dye. It does stain readily, and it's sometimes really, really, really hard to get out. So try to just keep it on the countertops. If you have to spill it, just spill it on the countertops because they're covered with absorbent paper, and it won't go through that. When you guys are doing your staining, leave the faucets running. That way the dyes and the chemicals are constantly going down the drain and being diluted. They're not just sitting in the bottom of the sinks, so they're just constantly being flushed down. This is the standard way of doing that, so just keep that in mind. You guys won't be using toothpicks, so you don't have to worry about this, but if you were, just make sure to discard them in a bleach solution. Okay. So there's a couple of important terms that you guys should know. There's a couple of different types of stains. One that we aren't going to be doing, but you do need to know about, is called a negative stain. It stains everything but the bacteria. It stains the background, usually kind of a black or purpley color. We used to use this a lot. It's very handy for bacteria that are really susceptible to heat, and so we can't heat fix these bacteria. They'll lyse. So we can actually just use this negative stain because it slowly evaporates its liquid and then just kind of locks into place. Because it's not staining bacteria, it's actually really easy to see on the slide. And we actually still use this all the time in science. The most important one is for electron microscopy. I'll show you guys that slide there. Yeah, I did. I'll just show you the slide. For microscopy, because what it's doing is it actually creates a thick layer that's electron dense, and that helps us visualize the edges of things in electron microscopes way better. So this is an electron micrograph right here. These are bacteria that are optically visualized through electron microscopy. This is actually protein. These are just little protein rings. Look at how that's really small, and we can visualize it on an electron micrograph because of this dye. So you can see those little donut holes right here. You can't really do that normally, but we can because we have that dye. So very handy, still useful. We're not doing it, so you guys don't have to worry about it too much. Just know that it stains everything but the bacteria. Simple stain. One dye to stain a bacteria just provides information about cell morphology and arrangement. So, hey, what shape is this cell? Is it a foci? Is it a spirali? Is it a bacillus? And what kind of arrangement is it? Staphylo, what have you. Differential stain. That's two or more dyes provide as much detail about the cell and its properties as you can. So in this case, we use it for like gram-positive versus gram-negative, or if it's producing mycolic acid, or if it has a certain protein on its surface. These are all really crucial things. So differential stains, mostly what we use in this lab, because differential stains give us a lot of information, and we can still get all the information that a simple stain gives us with a differential stain, and then some. So why not? Cut the middleman out, do extra stuff, just do everything at once. Okay. And you guys don't need to worry about negative stain, because you guys aren't doing it, so skip that part. We'll talk about gram stain. So it is the most commonly used differential stain in microbiology. It is actually one of the first steps if you send a clinical sample out they will probably tell you what the gram stain is of it. In fact, a lot of clinical samples will come back with that information on it at a bare minimum, mostly because it's so important. The only exception to that is if you're doing nucleotide sequencing, but that's a less common thing, but you guys will still see that a lot in reports. Developed by Christian Graham in 1884. So he basically kind of said, hey, this is the best way of doing it. He published a lot of papers. We were still kind of using this before, but we didn't have quite a good description of what was going on. He basically had all the information. So that's why I say that. Most important thing out of this, though, is that we have to use fresh cultures. The reason why is because fresh cultures give us the cells that are most intact. If they are older cells, they start to deteriorate. So gram-negative starts to kind of break apart. Gram-positive no longer holds on dye correctly. And so we see this thing called gram variability, where something's kind of purpley pink. That doesn't really tell you if it's gram-positive or gram-negative. You have to use fresh cultures. Fresh cultures give you the best chance at having everything that you need. So keep that in mind. Okay. Yes. Like 18 to 24 hours. Anything older than that, like a week old, is like going to give you really bad results. Okay. Peptidoglycan layers. So that's the protein matrix here. So gram-positive cell, nice thick protein matrix. Gram-negative, double layer cell membrane with a little bit of protein in the middle. So important that you guys are able to differentiate between gram-positive and gram-negative cells. The staining procedure, really simple. You have to memorize it. You have to memorize the times and what chemicals are used in it and what its purpose is, which is annoying, but I'm sure you guys can do it because it's only like 4 steps. You guys can definitely do it. The first one is crystal violet. Crystal violet is our primary stain. So whenever we talk about differential stains, we have a primary stain and a secondary stain. This one's crystal violet, nice deep violet color as the name implies. We have something called a mordant, which is a stain enhancer. In our case, it's going to be Graham's iodide. It's named after Graham, the guy who discovered gram staining. It's his recipe. It works actually the best. It creates a more intense, better purple, and that's the reason why we use Graham's iodine in our stains. Next one is our decolorizer. In this case, 95% ethanol. The decolorizing step is the most important step of any staining procedure. That is important to know. Make sure you guys memorize that. So everything else in here, you can kind of get away with being a little sloppy. Not this step. That is the most important step. You have to be very accurate with it. Last little one is safranin, which is our secondary stain or counter stain. It's going to basically stain all the bacteria of the other type in whatever stain we're doing. So counter stains are always the opposite of what the primary stain is staining. Little thing here, don't spill stain on the floor, on lab coats, on skin. Speaking from experience, it does not come out of skin very easily. These ones aren't terrible. They'll wash out within about a day or two, but it's not fun to have purple fingers or purple skin, so be careful with those. Okay. Oh, and I should mention, safranin, if you get it on your clothes and you use a detergent to clean them, it will actually degrade and turn brown. So you have a big brown splotch on your clothing, which is not fun, so don't spill safranin on yourself. Make sure you guys have your lab coats on. So what we'll do is we'll put crystal violet on the slide and let it sit for 1 minute. Sit there, chill out, absorb, glom onto the surface, and then we're going to gently rinse with DI water. So the key here is gently. You don't want to like blast the bacteria off. You want to have a nice gentle stream of water, and it will remove it. So even though this is not what happening in reality, basically you're lowering the amounts of crystal violet here. Now, if you guys remember the little thing we just watched about the current science, it's probably still on there on the gram-negative, but we'll just pretend that it washes away. Graham's iodine. Graham's iodine will form nice little crystal complexes. They like to form around crystal violet, and it will form a nice large molecule that's very hard to wash out. For the gram-negatives, it doesn't really penetrate in, so it just kind of goes away when you rinse it. The last step, most important step. I know it says 70%. I really got to fix that. There's different types, different percentages that you can use. 95% is just way better. 95% ethanol for 3 to 5 seconds. Now, this is actually a really key and important thing. 3 to 5 seconds is a range, which isn't very helpful for you, I know. But if you have a very thick smear, you might need to go that full 5 seconds. If you have a nice, good, thin smear that's easy to see through, then about 3 seconds will do. You don't want to leave it on there for a really long time. You don't want to be like, here, I'm going to stain like 3 slides, and I'll put ethanol on each of those 3 slides. You won't be fast enough. So only do 1 slide at a time. So you put the ethanol on there, let it sit for a couple seconds, and then you want to immediately rinse it off. You can over-decolorize the cells if you put it on there for too long. If you don't put it on there for long enough, you won't decolorize the gram-negative enough. So keep that in mind. There's kind of a careful balance point. It takes a little bit of technique to get it down perfectly. Last little bit is you'll counterstain with safranin for 1 minute. Now, because we've exposed this part to ethanol, these protein matrices are kind of all shut now, and they won't take up any more dye. So the safranin, even if it does stick on there, will mostly wash off. But even if it's on there, that's okay. Counterstains are almost always a lighter color stain, so they won't interfere with the primary stain. We use safranin a lot. Safranin is a very light pink. And you can see the gram stain will retain this. Now, another key feature about dyes is a lot of these dyes are charged. They have a slight positive charge on them, so they like to stick the surface of cells. And so bacteria still have a slight charge to them. They will stick onto those bacteria to a slight degree. That's why they'll retain that dye. And also, that's why negative stains are called negative, because they're negatively charged and repel from bacteria. Learn new stuff every day. Okay. So we've got all this, and now we've got our slides. We're going to look at it on the microscope, and we should see something that looks like this. Now, one step that you want to do after you guys finish your last rinsing step is you're going to want to blot it off with bibulous paper, which you found in your center drawers right next to your optical lens white paper. Key thing here, when you put the slide in there to blot it off, hey, don't just like wipe off the slide because you'll pull the bacteria off. They're not on there permanently, they're loosely on there, so you know, be gentle with patting it. You also don't want to pat it dry in the bibulous paper in your hand. So you just stick it in the booklet, you're going to put the booklet on the table and pat. Because your hand is curved, it's not perfectly flat, and you take glass and push down on it on a non-flat surface, it shatters, and you don't want shattered glass in your hands. So be aware of that. I say this because we've had people do this in the past, so be careful with it. Put it on the table, gently blot. You're going to put it in the microscope, and you're going to go all the way up to 1000x. So you have to use oil immersion for 1000x. And you'll see something like this. Now, these are examples of these. So Bacillus subtilis, Serratia marcescens, Micrococcus luteus. Very easy to tell. The gram positives are this nice bright purple. This one, because of the way it grows, it's not quite as bright, but you should get roughly the same kind of visualization. Serratia marcescens is gram negative. Gram negatives are much lighter. It's a little bit trickier to focus on them. But you guys should see them because we're making a mix slide. We'll be making a mix of all 3. E. coli, Bacillus subtilis, and Staphylococcus epidermidis. Even if you have a hard time visualizing the E. coli initially on the slide, you'll be able to see these other 2 and get it into focus, and then you can just hunt around for the E. coli. What you'll guys do is you guys will take your slide, and you will add a small drop of water to it, less than half a drop. So you have little bottles of water in the white trays at your tables. You'll a little half a drop of water. You'll take your loop, you'll streak the E. coli back and forth. You'll streak the Bacillus subtilis back and forth on it. And lastly, the Staphylococcus epidermidis. Most important thing here, a little goes a long way. You don't need a ton of bacteria. I'm certain that you've heard Dr. Lofton say, he likes to sing, keep the micro in microbiology. You just need a little bit of bacteria here. If you do too much, your smear will be way too thick and you guys won't see anything, which is really unhelpful. So make sure that you have just enough bacteria. On Lab Archives, there's there's a video that shows how to do smear prep and what a good smear versus a bad smear is. And you can see the bad smear is very dark and very hard to see through. The light smear is just enough bacteria to give you a good visualization, so keep that in mind. You let the slides air dry. Now, air drying takes a while, maybe 5 minutes. If you put too much water on there, it's going to take like 10 or 20 minutes. You can speed it up in one of 2 ways. Actually, one of 3 ways, but we'll tell you the first 2 because those you can do freely without your TA's help. The last one you need your TA's help. The first one is you can take a stack of paper towels and wave it back and forth and help the air move across it so it releases the water a little bit quicker. You can also take a hot loop, flame it, let it cool down just a little bit, and spread the water back out, because sometimes the water doesn't like to stick to the glass very well. So it kind of globs up on itself. If you spread the water out, it's got a larger surface area, so it evaporates more quickly. The last one, especially if you have way too much water on there, it's taking a long time, you can go see your TA and they will help you put your slide on a hot plate that's set to a very low temperature, and that will help it evaporate faster. That's not an optimal thing because if you do it too long, then it fries the cells and they will not work right on the slide. So keep that in mind. That's like worst case scenario situation. Try to do it with the air drying first. That's much more gentle. After you guys do that, you guys will follow the Graham Staing protocol, which we kind of talked about already, so we'll go over it again. Also in your lab manuals. Once you get to the end of that, you will blot the slides to remove any liquid and excess dye, and then you guys will look at them on the microscopes and take a picture of them. This one is important to do, the acid fast, because it is the foundational way in which we differentiate bacteria. And it will become very important later on in the semester when we do our unknown identifications, because we're going to be doing them with Graham staining as our first differentiation technique. So make sure you guys really get this down. Don't kind of gloss this. It is very important to know how to do. Does anybody have any questions about Graham staining so far? Make sense, kind of? Okay, your TA will help you guys do some demonstrations of some things, so keep that in mind. Okay, we're going to talk about the other two stains that we will do on Thursday, which is acid fast, also called the Zeal-Nielsen method. You guys should know that. Write it down. And the reason why I say it's important is those guys kind of figured it out and they had a really hard time doing it, and it's kind of a very good standard. Now, important things here. We will be using some BSL-2 organisms in this one, so be careful with that. Be careful when handling the culture tubes so you don't drop or break them. Wear goggles, gloves, and lab coats all the time. Do not heat empty beakers. We will be using hot plates to heat up water for steam baths. Don't let them run out because hot glass that's empty can shatter, and then you don't want flaming hot glass shooting everywhere. So please be careful with that. All staining must be done under the scavengers, which are those little white PVC elbow things that are sticking out of every table in the back. You got to be underneath that so that it's constantly sucking up the vapors. In part because we're going to be using a phenol-based stain, which evaporates very quickly, and because we're adding heat, it evaporates even quicker. That's not a word, but still keep that in mind. Now, of this, you should know, because it's phenol-based, some people are very sensitive to it, and it can give you a headache. If you start feeling lightheaded or like not well because you've been inhaling the fumes because you've maybe been hanging out a little too close and not having the scavenger right over the top of it, one of the best things you can do is just say, take your gloves off, wash your hands, leave the lab, and just stand outside for like 2 or 3 minutes and take big, deep breaths in, and it tends to make people feel a lot better. You just kind of have to let that phenol get out of your system. Now, of note for that, very important, you don't want to have any dye get into water, and I'll explain why in just a second. When you guys are done, make sure you guys turn the hot plates off. Use pot holders, remove hot beakers from the hot plate, clean out your beaker when you're done. Remove gloves when you are using your microscope so you don't get any dye on the microscopes. So what are acid-fast bacteria? These guys have something called mycolic acid on them, which is these chemicals right behind here. They form this nice, really long, thick layer. And they are very waxy in nature. They're non-polar. Because they're non-polar, they resist letting charged things go through them. In fact, it's a very, very clever method of helping themselves stay away from your immune system because not a lot wants to bind to non-polar things. So they are very, very good at resisting staining because of this. So to do this, we have to use heat to force the dye through. We also used a phenol-based dye, and phenols have a very large non-polar region that helps them move through this mycolic acid membrane much easier and get trapped in there. Once you get the dye in there, it becomes locked in place and it will not let up that dye even under acid conditions. That's the name acid fast. If you guys have ever done any like archival work or something, you might have heard of acid fast ink and paper. Same kind of principle here. It's just something that's resisting staining, destaining, decolorizing through acid. Okay, so what bacteria have mycolic acid? Well, actually everything in the Mycobacterium genus. So every species there is going to be an acid-fast bacteria. They are all aerobic gram-positive small bacilli. They're really tiny typically. They form dry, crusty growths on average. They are present in things like soil and water. Some of them are pathogens, which we'll talk about in a second. We'll be using a strain called Mycobacterium smegmatis, which is a BSL-2. It's found in soil and water. The two major diseases you have to know about for this one are Mycobacterium tuberculosis, which causes tuberculosis, as I'm sure you can guess, and Mycobacterium leprae, which causes leprosy, which I'm sure you can also notice. Now, Mycobacterium tuberculosis. There's a number of ways we can test for this. The most common one is a thing called the mannitose skin test. You guys do have to know about this. Make sure you guys remember it. You guys have probably done one, potentially. All you do is you take a little bit of antigen, you scratch your skin. If a welt forms from that scratch, that means that you've got a response against that antigen, which is the Mycobacterium antigen. Now, it doesn't mean that you're sick, but it's a good indication that you might be. And the reason why is because if you've recovered from a tuberculosis infection, you don't have tuberculosis anymore, you'll still show up positive on the mannitose skin test. Or if you've had the BCG vaccine, you will also show up positive on the mannitose skin test. Or sometimes your body just reacts really badly to the mannitose scratch skin test. And you show up positive for no reason at all. I know this because my wife's father, way back when he was a kid, got a mannitose skin test and showed up positive for tuberculosis, and they quarantined him for 3 weeks. And this was a no kidding around, like, you have to be locked in your house, and people from the state health agency will come and check on you every single day, kind of thing. Turned out he didn't have tuberculosis. He just had a reaction to the type of test. So he got quarantined quite unfunnily for that. Now, when you show up positive on this, the next thing they're going to do is they're going to do one of two things. They're going to do a chest x-ray, and they're going to look for tubercules. They're also going to look for sputum to do a sequencing on to see if it's tuberculosis. Now, sequencing, not considered a gold standard. What we still do to this day is culturing. And culturing is done on this egg-based media, which smells horrific, so be glad we don't ever have to do this. And the downside to this guy is that it takes about 3 to 6 weeks to grow. It is an exceptionally slow-growing bacteria, and it forms these very hard little tubercules in the lungs. So that's what it looks like when you cut a lung in half, it has it. So they're very slow growing. So the patient could die in that 3-week period, so you don't want to wait around for it. So the fun thing is we can do an acid fasting on these and get a presumptive diagnosis, because if you're going to have mycobacterium in your lungs, it's probably going to be this guy. Now, it could actually be a couple others, but mostly this guy, and we kind of treat them the same way. So the last one is Mycobacterium leprosy, or leprae, which causes leprosy, also called Hansen's disease. It does a lot of wacky things to your immune system. It creates these large vacuoles and macrophages and things like that. Contrary to popular opinion, it does not make your limbs fall off, but it does cause ascending loss of sensation in your extremities, and it can go all the way up to the trunk of your body. And if you can't feel your hands or toes and you injure them, the likelihood of you getting further infections is really, really high. And because you're getting these constant tissue infections that you can't take care of, and you constantly keep damaging because you can't feel your fingers very well, then the easiest thing is sometimes just to cut those fingers off, or they'll just start degrading because of all the damage that they receive. Completely treatable disease. We still have a couple leper colonies in the world, but basically we can just treat it with antibiotics and cure it. So typically the places where it's still around and kind of prevalent is places where it is so socially unacceptable to have leprosy that people try to hide it. And that lets the disease progress really far, and then they have a severe problem rather than just seeking help and then they've gotten treatment really quickly. Okay, now our non-acid fast bacteria is going to be this stuff called Staphylococcus epidermidis. Most of you guys have this on your skin. That's the name epidermidis. It's on your epidermis. It is a gram-positive foci. It makes these nice little white colonies. It is a non-acid fast bacteria, so it will not take up that dye very well. It'll let go of the dye really easily too under acid conditions. One of the but interesting things, it's most commonly known for nosocomial infections in certain situations. So it is considered BSL-1, so it doesn't typically cause disease in humans, but sometimes it can. And the most common one is the leading cause of intravascular catheter infections, cath lab infections, and 40% of valve prosthetic endocarditis, where basically an infection happens and it hardens the pig pericardium that they use to make these artificial valves with, and that's what it looks like. So this is what it's supposed to normally look like, a nice little white membrane on a little wire mesh thing. And this is one that they took out of a patient that had a severe thing where it just ate away at the tissue and it hardened, crusty. That's obviously wrong. So there you guys go. procedure you guys are going to do is you guys are going to mix together both Mycobacterium smegmatis and Staphylococcus epidermidis on a slide. You're going to let it air dry, heat fix it, and then you guys are going to stain it with your primary stain of carbonyl fusion. And you're going to keep reapplying the dye as needed for 10 minutes over a steam bath because you've got to help that dye force its way in there. You're going to then decolorize with acid alcohol. You're only put it on there for a couple of seconds and then rinse it off really quickly. And lastly here, we're going to counter stain with methylene blue. Now, methylene blue, we don't have to counter stain with heat. And the reason why is because we're staining bacteria that don't resist staining. So any bacteria that doesn't have the mycolic acid will just take off the methylene blue, no problem. So methylene blue for 2 minutes, rinse it off, and then you guys can view it. And anything that's acid fast is going to be this nice red-violet color. So you guys can see the bacteria sitting here in these nice little dense colonies. And then the non-acid fast, the blue ones, are these nice little blue-looking guys right there. Now with this, very important for applying the stain, this dye is very drippy. And like I said, it is phenol-based. So if you accidentally get it in the water, it's going to aerosolize quickly and give everybody in the lab a giant migraine. Don't do that. That's bad. The way you can avoid doing this is while they're over the beakers, you will apply the dye over the top of the slide. And so when it drips onto the slide, it's just going onto the slide. It can't get in the water. What you don't want to do is go from the side over the water, because if it drips out while you're going over the water, it gets in there, and then we have to dump all the water out and you give everybody a lot of headache. So don't do that. That is bad. I'll also warn you guys that the dye does like to soak through nitrile. You've got a couple of seconds of resistance on it, but because it's very good at kind of passing through things, if you get a bunch of phenol on your hands, you usually want to take your gloves off and change them out. And the reason why you want to do that is because if it actually gets on your skin, it'll get in the bloodstream and it'll give you a headache for like 3 hours. It's not fun. Speaking from experience. Now, how dry is dry and how wet is wet? Well, a wet one should look like this and a dry one should look like this. I know it's kind of hard to tell, but it should kind of have this moist quality to it that's kind of shiny for the wet one. And the dry one will just kind of have this like kind of dried up look to it. It won't have that glossy texture to it. If it changes colors, sometimes it turns slightly greenish. Don't freak out. That's totally normal. As long as it has that kind of wet look to it, then it's fine. But you're going to have to reapply dye repeatedly. It's going to evaporate kind of quickly. Okay, other thing. How to do the smear. Smear's got a little bit of technique to it. You want to have a slight overlap. And the reason why is because the Mycobacterium is not very proliferous, and the Staphylococcus aureus is very proliferous. So there's a ton of it in any sample you put on there. It will kind of overwhelm the Mycobacterium. You'll still be able to see them, kind of, but it'll be really hard to differentiate. So if you have just a slight overlap, you'll definitely be able to see some Mycobacterium all by themselves on the left side, and you'll only be able to see a couple on the, definitely be able to see the Staph aureus on the right, Staph epidermidis, I should say, and in the middle, you can see both. So just keep that in mind, just a small overlap. That's the best way to do it. Okay? All right. Next stain, last stain, then we're done. How are we on time? We're great on time. Okay. Schaefer-Fulton method. Endospore stain. Schaefer-Fulton is the method. Endospore is the stain. Remember that acid-fast is Zeal-Nielsen. These guys are Schaefer-Fulton. Same kind of setup as before. Notify your instructor if any science or culture breaks. Malachite green will be used. It aerosolizes quickly and will get on your clothes and face, which is not fun. I'll talk more about that in a second. Always perform this procedure under a scavenger and use goggles, gloves, and lab coat at all times. Do not eat empty beakers. Make sure you dispose of everything in a proper place. Now, malachite green also aerosolizes really quickly. And it does not cause a headache, but it does, the dye itself does aerosolize. And the dye itself will stain things in its aerosolized form. So if you were standing over the beaker watching it, it will stain your forehead a nice little tinge of green. And if you have very light hair, it'll stain the front of your hair, slight tinge of green. And that sounds funny, except it doesn't come out for about a week. It's a very long-lasting dye. So don't do that. It's a bad idea. Look at it from a distance and don't put your head over it. Okay. Sporulation. So when we're talking about spores, sporulation is the process of forming spores. And typically this happens when conditions for the bacteria are bad. Hey, we can't survive, we're dying here. we need nutrients, or we're getting bombarded by antibiotics or UV light. So let's shut everything down, hold out a little cell, a spore, and we can survive. And so it's just a survival mechanism. They can't reproduce as spores. They're just sitting there chilling, waiting till conditions are good again. They're resistant to heating, drying, freezing, UV radiation, chemical agents. That's a whole lot of things. The only way to really kill them is with high energy radiation, so gamma radiation is kind of the common one. During sporulation, cells will undergo changes to encapsulate DNA ribosomes within a central core, and there's actually been a lot of research done on this because it's a really neat and interesting survival mechanism. Some words you guys have to know. Sporangium. The sporangium is a vegetative cell, so it's a pink cell, nice, healthy by itself, with a spore inside of it, so an endospore. So sporangium has to be both of those things. The endospore is retained within the sporangium. It can be located kind of like either in the very center of the cell or on the kind of edge of the cell, which is called terminal, or subterminal, which is kind of like on its way to the edge of the cell, so in between the end and the middle. And lastly, it could be a free spore, so it's not really an endospore, it's a free spore. It doesn't have any cell around it. It is just out in the environment by itself. That's another option as well. Germination is the process by which free spores revert to a metabolically active cell. This happens when the environment is good again. So once things got, like, it's got nutrients, it's no longer getting bombarded by UV radiation, it'll start growing again. Sporulation and germination are not methods of reproduction. They are for ferns, they are not for bacteria. Ferns have shockingly similar language because biologists hate learning things. So this is, just remember, not reproduction for bacteria, just survival mechanism. Okay, there are 3 major genuses that form spores. You do have to know these 3. They're really easy to memorize. Bacillus, Clostridium, and Clostridoides. Clostridoides is new. It's not really new. We just kind of said, it's too different from Clostridium, so we made a separate one. Clostridial organisms. They are all strict anaerobes. They're found in soil. They're found in dust. The one that you guys are going to be familiar with a lot is probably Clostridium botulinum and Clostridium tetani. Botulinum causes botulism. It produces a toxin, a neurotoxin, which causes flaccid paralysis. If you have ever seen a canned good have like a big bulge on the side, that's a pretty good indication that it might have botulism in it because it produces a lot of gas as a byproduct of its anaerobic fermentations. Because they form spores, if you don't can properly, don't bring it up to a high enough temperature, the spores will survive and they'll start causing that bulging. So that's why canned good can sometimes cause that. Now, you guys might know that the protein for it, the toxin, is used for Botox, which helps get rid of wrinkles. We've actually been using it for a lot of other things. There's actually 2 other clinically approved uses for it that were actually before using it for cosmetic purposes. Does anybody know what the original uses for Botox were? You have a guess? Right, but before that, it was used for something else. Yes? Migraines, yes. So facial pain, muscular contractions on the face would cause migraines. And so to help relieve those muscles from contracting, they would inject them with this toxin to help release it. And then the people were like, hey, look, this gets rid of my wrinkles because now my face is relaxed. And that's the beauty part. Before that, the other one is for excessive sweating. You can inject it into your palms and in your feet, and it actually reduces, and your underarms, reduces excessive sweating. So if you have conditions where you're sweating a lot from your hands and feet, you can actually paralyze it for something like 3 to 6 months. Very handy. There's also a new one. I don't know how much it's still in use, but they also did one where they would, for people that have cluster migraines, which are located deeper in your brain, and of course that's harder to treat, they would take the needle and inject it through the roof of your mouth up near the base of your skull, inject the toxin in there. It would actually relieve cluster migraines for like 9 months. You know, and of course the only downside is you have to have a giant needle shoved up near your brain and injecting yourself with neurotoxin, but it did actually work quite well. The other one is Clostridium tetani, tetanus, lockjaw. It's a neurotoxin as well, but it causes the exact opposite. Uncontrollable muscular histotenosis, so clenching contraction of skeletal muscles. It can be deadly. It can prevent you from eating or just cause you excruciating pain. That's not very fun. Of course, there's a vaccine for it. The vaccine is against the toxin, not against the bacteria. In fact, your body can clear out the bacteria, no problem. Toxin is what we're worried about, and so all of your immune response from the vaccine is actually towards the toxin and helps get rid of the toxin produced by the bacteria. Okay, Clostridium. So this one is also a strict anaerobe, and it's found in the intestines of mammals. And there's only 2 organisms within this. Only one that's really clinically important, which is Clostridium difficile. Yeah, everybody says C. diff. Okay, but I'm actually going to show you guys because I asked a Latin professor. I was like, "Hey, how do you say this?" Because I had no idea. He said that the most likely pronunciation would be Clostridium difficile. So if you guys want to sound fancy in front of people and you're like, "Hey, I know how to say C. diff full way," there you go. Highly contagious gastrointestinal disease. Because it's a spore former, it's very hard to get rid of. It is exceptionally resistant to antibiotics. And it doesn't seem like it would be that dangerous, but it actually is, mostly because the populations it affects the most, which are people in elder care homes, because they often share rooms with each other. Their immune systems are more compromised. They're much more susceptible to getting sick and being stationary and thus getting secondary infections and dying. About 10 to 30% of patients that get it will get a recurrent version of it. And the recurrent version is almost always worse than the initial occurrence of it. And the reasons for that are many, and we're not going to get into those. But you should be aware that that's actually really serious. About 20% of people die from the infection overall. If you get a recurrent infection, your likelihood of dying goes up to 36%. That's a huge number for a gastrointestinal disease in the modern era. And these are not like, hey, this is what the death rate is in a country that has not a strong healthcare system. This is US, Europe, standard numbers all across modern healthcare systems. So it is a serious disease, believe it or not. Okay, last one is bacillus genre. There's a bunch of bacillus-based diseases. They're all large gram-positive rods. We're still good on time, but I'm almost done here, so don't worry about that too much. They can either be aerobic or facultative anaerobes. The really interesting ones that you guys probably know of that cause disease are Bacillus anthracis, which, as you can guess, causes anthrax. There's actually 3 different types, but you guys didn't know that. There's a systemic type, there's a gastrointestinal type, and there's a pulmonary type. That's not going to be on the test, but you should just know that there's different types. One of the kind of key ones that you see typically produce black lesions, and it's not really a symptom after the fact, but if you die, the corpse will lack rigor mortis. So most corpses, after a certain amount of time, will resist being moved until they've decayed enough to be able to be movable again. These ones, when they die, will lack rigor mortis fully. They'll never have that stiffness to them. So weird. Bacillus cereus, on the other hand, is the most common source of food poisoning in America. It likes to grow on starchy foods. If you keep it at the wrong temperature, you'll get it into the danger zone. That's the one that gets replicated on there and causes the most poisonings. So the way we do this endospore method is we take a slide, we put some bacteria on that, and we're going to expose it for 5 to 10 minutes. 10 minutes works out better, so I'm just going to say 10 minutes. If you guys get rushed on time, you can go down to 5 minutes. But with your TAs helping you guys, it should be about 10 minutes. We're going to be applying malachite green. Same kind of thing. You kind of have to reapply it so that it stays nice and moist. After 10 minutes, you're going to take a little paper towel off and throw it away in the trash, and then you're going to decolorize with water. Decolorizer here is water, not alcohol or anything else, just water. You're going to counterstain it with safranin for 1 minute. So if you guys remember, safranin is what we use for gram staining. We're going to use it here as well. We're going to be using Bacillus megaterium cultures that are about 72 hours old. Now, remember I was talking about fresh cultures before for gram staining? We actually want old cultures here because we want to stress out the bacteria so they form those spores. If they're not stressed out, they won't form the spores. You'll just see normal, regular-looking bacteria. So we stress them out a little bit. So they're like, I can't survive. And so they'll start producing the spores for you. So keep that in mind. Okay. Clean up stuff. Same kind of thing. Make sure you guys unplug the hot plate when you guys are done. Rinse out the beaker when you're done. Remove all labels from tubes. Put them in the autoclave cart. You guys don't have to worry about cleaning your slides. Just ignore that part. Put the microscope back in the cabinet where it goes. And make sure you guys submit your report by the end of the day. Now, before I let you guys go, 2 things. 1, we are going to start checking for one safety issue that's kind of persistent and people tend to forget about. Pant leg length your pant legs have to go all the way down to where your socks or your shoes are. If you have a bunch of exposed skin right by where your ankles are, that's kind of a problem because if something's going to spill and break, it's probably going to do it on the floor, and you don't want a bunch of exposed skin where that happens. So double check before you leave the house on Tuesday and Thursday that you have full-length pants. Lastly, does anybody have any questions? All right, I will see you guys next week. Have a safe weekend. Very safe because it's holiday weekend. And I will see you guys later.